Howdy, folks. Thanks for checking into Mr. Ulrich's landofbiology.com. I am Mr. Ulrich. In this second of our trio of notes casts on cellular respiration, I'm going to be focusing on the Krebs cycle. If you haven't watched the first video on glycolysis, you should probably do that. I'm going to review a little bit here in the beginning, but to get the full picture, you really need to watch the first video. In the last video, I discussed with some detail the oxidation of the 6-carbon sugar glucose into 2 3-carbon pyruvate molecules. And one of the hallmarks of this anaerobic pathway is that it is inefficient. For each glucose molecule, we're only producing a net of 2 ATP molecules. And that's because pyruvate still has 3 carbons bonded to each other, and those bonds represent some chemical potential that still could be released if we could only break it down some more. If oxygen is available, and of course we're talking about a eukaryote here, that pyruvate then can move into the mitochondria, where the Krebs cycle is going to oxidize it even more, breaking it down from the 3-carbon pyruvate into 3 single-carbon carbon dioxide molecules. To look at a general overview here of cellular respiration, this diagram is a really good diagram because it shows not only the 3 main processes of cellular respiration, but also where it happens and what the primary energetically important products are of each one of those stages. Where glycolysis is taking place in the rather formless soup that is the cytosol or the cytoplasm, the rest of aerobic respiration is going to be taking place within the structure of the mitochondria. So we should probably talk a little bit about the structure of the mitochondria itself. I describe it as a small bag with a big bag shoved inside of it. And so the outer membrane is going to be smooth because it's stuffed full of inner membrane. That inner membrane, because it's bigger, has to be folded in order to fit. Each one of those little folds is called crista, plural, it's cristae. And what this really sets up is a space between the inner and the outer membrane, which we conveniently refer to as the intermembrane space. It is fluid filled. It also, the innermost chamber of the mitochondria, and within that inner membrane is called the matrix. It is fluid filled. Floating around in there are the mitochondrial DNA, a single loop of DNA, and mitochondrial ribosomes. They have their own ribosomes. Also in there are the enzymes that are synthesized at the ribosomes of the mitochondria from the mitochondrial DNA. Many of those enzymes are floating around in the matrix, where also many of them are embedded within the inner membrane itself. The mitochondria, we refer to them as the powerhouse of the cell. We know that's not where all of the ATP is produced, but it's where most of the ATP is wrung out of the glucose molecule. So when we start thinking about cells that are going to have lots of mitochondria, yup, you heard right, mitochondria have their own DNA. They have their own ribosomes. They make their own proteins. In fact, heck, they divide on their own. A lot like bacteria do. Lynn Margulis took all of this data, all this information, and formulated what she referred to as the endosymbiotic theory, talking about how mitochondria and chloroplasts were once free-living bacteria on their own, developed a symbiotic relationship, and then became completely dependent upon their host cells, and the host cells completely dependent upon them. Also, So in there that allows the mitochondrion to do its job are these membrane-bound proteins, these enzymes and permeases that are going to make up an important part of the electron transport chain, which we're going to get to in the next video. But right here, we can mention that having that highly folded inner membrane is going to allow for more inner membrane, hence more of these membrane-bound enzymes and permeases and proteins to do the job of electron transport that we're going to talk about, like I said, in the next. The first thing that has to happen is we have to kind of prepare the pyruvate for the Krebs cycle. All right, so the pyruvate is floating around in the cytosol. It's going to make its way through a transport protein right into the matrix where there's lots of coenzyme A. That pyruvate is going to be combined with the coenzyme A to make acetyl coenzyme A. Now, the problem, of course, is that pyruvate is a 3-carbon and acetyl coenzyme A is a 2-carbon molecule. So what has to happen, of course, is we have to strip one of those carbons off, and we do that in the form of carbon dioxide. But this is oxidation, and oxidation needs to be coupled with a reduction. For every oxidation, there is and also a reduction. So here we're going to get that NAD to come in, pick up a hydrogen, and be reduced to NADH, because that hydrogen is going to bring an electron with it. Here we are, halfway through the video, and I'm just now getting to the Krebs cycle. So who is this Krebs guy? Well, Hans Adolf Krebs was a German-born British researcher who back in the early 50s won a Nobel Prize for determining the 8 steps of the citric acid cycle. Each one of these steps is catalyzed by a different enzyme, and it is not important for you to memorize each one of the steps. I show you all this stuff again so that you can see that it's not magic, that it's chemistry, and that it can actually make sense. What the Krebs cycle basically does is it starts with a 6-carbon citrate molecule, and that gets stepwise oxidized down to a 4-carbon oxaloacetate molecule. And if that doesn't make any sense to you, that makes sense because we haven't talked about the rest of the cycle. It did evolve to come about later than glycolysis did. It has to, because it starts with the products of glycolysis. So glycolysis had to be figured out evolutionarily before the Krebs cycle and the rest of aerobic respiration. Bacteria have been doing this glycolysis business for a long time, and then along came some bacteria that figured out how to go through photosynthesis, and in doing so started to fill the atmosphere with free oxygen. And the availability of oxygen is what makes aerobic respiration. Being able to account for the carbons is a good way to introduce the Krebs cycle itself. So we start out with that pyruvate, and that gets oxidized down to a 2-carbon acetyl coenzyme A. And this acetyl coenzyme A is what is going to enter the Krebs cycle itself. All right, now remember, this is a cycle, so some of those carbons are going to just keep getting recycled around and around and around and around and around. All right. The first thing that happens is we add that 2-carbon acetyl coenzyme A to the 4-carbon oxaloacetate molecule to make the 6-carbon citrate molecule that begins the Krebs cycle. That 6-carbon citrate molecule is then oxidized down to a 5-carbon molecule. And that, of course, means that we're going to lose a carbon dioxide. It gets further oxidized down to a 4-carbon molecule, releasing another carbon dioxide molecule. Those 4 carbon molecules then go through a number of changes until they become oxaloacetate, which can then be bonded to another acetyl coenzyme A, and around the horn it goes. So each time there's a glucose molecule, you're going to get 2 turns, because there's 2 pyruvate molecules, 2 turns of the Krebs cycle. We should also be able to account for the electron carriers because they're going to become very important in the electron transport chain. So when we oxidize that 6-carbon citrate molecule down eventually to the 5-carbon molecule, yeah, we're going to lose some carbon dioxide, but we're also, because we're oxidizing, we also need to reduce something. So we're going to make some NADH. In fact, every place where we lose a carbon dioxide, then we're going to have to make some sort of reducer. Or a reduced species. Now, we're also going to make some ATP. Woohoo! Finally, we get some energy out of this thing. All right, so we make one ATP molecule. But we also are going to make another electron carrier, one that we haven't talked about, FADH2. Right? Does the same job as NADH, but it's just a slightly different form. All right, we're going to make some more NADH in one of the transformations. Heck, we're going to make some NADH right in the beginning when we strip that first carbon off of the pyruvate. So each time we go around here, we're going to make quite a few reduced molecules as well as an ATP molecule. To look at glycolysis and the Krebs cycle together, we start out with glucose, and we fully oxidize that down to carbon dioxide. In doing so, we're going to yield a total of 4 ATP. We're going to net 4 ATP. Remember, we get 2 ATPs from glycolysis. And then we get 1 ATP out of each pyruvate molecule here. And since there are 2 pyruvate molecules, that's 4 ATPs total. Still not a lot of ATP. Still not enough to run a big clunky multicellular eukaryote like an animal. Well, if we're not getting a whole lot of ATP out of glycolysis and the Krebs cycle, then what's the bonus? What are we actually doing it for? Well, that Krebs cycle is going to produce all of those electron carriers. Remember, anything that's going to carry a hydrogen is going to carry the proton and the electron. So we are shuttling electrons around every time we shuttle that whole hydrogen molecule. So we're making lots of NADH and FADH2. And those are going to go, like I said, to the electron transport chain. It's in the electron transport chain that we meet our friend ATP synthase, which is going to do the big job of making a whole lot more ATP than either the Krebs cycle or glycolysis are doing. Some quick accounting here of the energy in the TCA cycle. We start with that pyruvate molecule of 3 carbons, oxidize that down to 3 1-carbon carbon dioxide molecules. We'll make some some NADH and some FADH2 molecules. Save those for later on. And then we'll also make some ATP, hooray! We have to remember that because we're dealing with 2 pyruvate molecules from each glucose molecule, that all of our yields get doubled when we start thinking about per glucose. So we get a net gain of 2 ATP molecules, and we also get 8 NADH and 2 FADH2 molecules, which we're going to send off to electron transport chain. So what's the value here if we're only getting the 2 ATPs? We're getting those NADH and FADH2 molecules. All right, they're carrying electrons along as hydrogen molecules. So we get to move those electrons. We're also going to be moving the hydrogen nuclei, the hydrogen ions themselves. So they get moved and they're going to become important here later on in the electron transport chain itself. So all of those NADH and FADH2 molecules, though they're not really usable for energy by the cell, they're important for creating energy that's going to be usable by the cell. Again, what's the point of cellular respiration? The point is to make ATP. We still haven't made a whole lot of ATP yet, have we? So how are we going to make all of those ATPs? We've only made 4. We need a lot more in order to run big clunky animals. So how are we going to do this? Well, this is where ATP synthase comes in. ATP synthase is a embedded membrane-bound enzyme. As a hydrogen ion gradient is set up between the matrix and the intermembrane space, that's going to cause a difference in concentration is going to cause the diffusion of those hydrogen ions. They're going to flow through that ATP synthase, and as they do, that causes the ATP synthase to spin. All right, and that conformational change is going to mechanically force an inorganic phosphate molecule to bond to an ADP molecule and make us some ATP.